Our second student presenter, Danielle Vansover. Danielle's a senior at New York University, where she's majoring in biology. Danielle's passionate about aging research because she seeks to define the absolute truths of biology. I know she's learned a lot this summer about how the passage of time might be absolute, but diseases and processes of aging are not. Danielle's previous experience focused on regenerative medicine and mesenchymal stem cells contributions to skeletal muscle and adipose tissue. Danielle's interest in regenerative biology was a perfect fit for her summer placement here at SRF under the advisement of Dr. Hadi Riba. Danielle will be speaking today on heme, the toxin in our blood, investigating heme toxicity in an aging context. Thank you for your time, Danielle, and I can't wait to hear more about your work this summer.
All right. Hello, everyone. My name is Danielle Vansover. As Lily said, I'm currently an undergraduate at New York University, and I'm studying biology. And today I'll be sharing my research from this past summer as a scholar at Sense, investigating heme toxicity in an aging context. So to provide a bit of background, I want to briefly start off with what it means to age. So the phenomenon of aging, looking at it through a biological context, is often generally defined by the buildup of damage. Over time, our bodies undergo what could be described as wear and tear. The many physiological systems and intricate mechanisms key to our survival gradually decline in function. And in the face of many pathologies and diseases, this decline only accelerates. And the sources and symptoms associated with this decline include many familiar phenomena, such as genomic instability, telomere degradation, epigenetic changes, and many more. So there are many factors that contribute to the aging process. One such factor investigated heavily at Sense is that of senescence. It's widely understood that our cells undergo a variety of changes, and this is generally dependent on the locale of a cell and tissue state. And senescence is a particular cell fate of great interest, as it's found to be highly associated with aging. As we age, senescent cells increase. A senescent cell is one that no longer divides, yet it still remains alive, often described as a zombie cell. Senescence does have a necessary function. It protects against uncontrolled cell growth, as seen in cancer. Yet, in spite of this function, senescent cells also resist the body's usual system of disposal and often linger in their environments where they secrete high levels of SASP factors. These are harmful compounds that cause further damage to surrounding tissue. And this summer, I had the opportunity to work under the mentorship of Dr. Hadi Reba with the Replenisense Lab. And a big question that Repleni is investigating is not necessarily how we can remove senescent cells, but whether we can actually delay the onset of senescence in the first place, and therefore maintain a cell's functionality and capacity for longer. Now, this would have to apply to the many processes that occur in the body, all of which possess many distinctions from one another and would first need to be comprehensively and precisely defined. So currently, the lab is investigating one specific phenomenon that is highly relevant and occurs in all of us on a regular basis, and that is hemolysis. And the lab is studying this and the many implications associated with it as significant sources of damage and aging. So hemolysis is the destruction of red blood cells. It's a completely normal, steady state process that occurs in all of us, since the body needs to constantly replenish old and damaged red blood cells. Now, a single red blood cell contains approximately 260 million hemoglobin molecules. These are the pretty well-known proteins responsible for oxygen transport through the bloodstream. During hemolysis, however, these hemoglobins are released from the cell, at which point they can be oxidized or broken apart, and in turn, they release a compound known as heme. And so heme, it's an essential compound that performs many functions associated with gas exchange, energy production, and signal transduction, among other functions. Yet, when completely exposed from the red blood cell and no longer housed within a protein, Heme has been found to be highly toxic due to its hydrophobic properties and high reactivity. It promotes oxidation, changes the structures and functions of essential proteins, and generally causes significant cell and tissue damage. And in the case of many blood conditions, including sickle cell anemia, thalassemia, and even during blood transfusions, hemolysis is upregulated, and red blood cells are actually destroyed much faster than they can be replaced. And in these cases, the free heme pool that's established continues to accumulate, and its effects become increasingly severe. So, the central question that my project is hoping to answer is, can we determine whether heme is a source of cell damage and senescence? And if so, can we then develop a targeted therapy to reduce free heme in the body and ultimately minimize its effects? And so, in order to begin addressing these ends, my project focused on two central aims. The first is to test heme toxicity on cells of interest, observing both the short-term and long-term effects. And the second aim of this project was to test certain compounds as potential sources of heme neutralization. I focused primarily on albumin, the most abundant protein found in the plasma, and therefore one that would very likely interact with free heme in a hemolytic event. And albumin is known for its antioxidant properties, so a goal was to test those in cells against heme. So to test the short-term effect of heme and the protective properties of albumin, IMR90s and HUVECs are cells of interest, were cultured and then treated with heme and albumin in a dose-dependent manner. At the endpoint of a treatment, cell viability was measured using a luminescent ATP detecting assay. So essentially, a greater signal indicated a greater amount of metabolically active cells. So these are the results of this experiment in IMR90s. In the two sets of images on the left, we can see the two extremes of heme treatment with no added albumin. So the top image depicts healthy cell proliferation and extremely minimal cell death. And in the image below it, containing 10 micromolars of heme, there's an evident reduction in cell survival in comparison. And the four images on the right all contain 10 micromolars of heme, yet they also contain increasing amounts of albumin. And as we can see, with no albumin included, cell survival is significantly reduced, yet already at 25 micrograms per ml of albumin, proliferation picks up, and by 100, cell survival is nearing the amount seen in the original control. And these two graphs depict these results. The graph on the left displays this treatment in IMR90s, and on the right in HUVECs. An initial view of these gathers that as heme does increase, there's a reduction in cell survival, yet adding albumin treatments in an increasing manner to all heme doses appears to allow cells to recover. So this is a positive sign for albumin and begins to confirm that it has some protective properties against direct heme exposure. The second aim was to determine the long-term indirect effect of heme. Part of heme's toxic repertoire is its ability to oxidize proteins, which then inflict further damage to surrounding systems. And to model this, albumin was selected as the protein to be oxidized due to its abundance in the plasma. And two different oxidation methods were attempted. The first used copper sulfate and the other hydrogen peroxide and heme. After incubating the protein with either compound, oxidation was verified using a detection kit, and these samples then underwent immunodetection to be visually confirmed. This was perhaps the most challenging aspect of this project, since no oxidation protocol had been previously set. So there was a lot of trial and error that went into this endeavor. but it was a welcome challenge, and I definitely learned a lot from it. So in terms of results, these depict one such attempt at using hydrogen peroxide and heme to oxidize albumin and subsequently treat IMR90s with the oxidized protein. So the Western blot image contains samples of unoxidized and oxidized albumin with and without the use of the detection kit. And while some differences are observed in the oxidized sample, I'd conclude that these results are far from final and that a more optimized protocol be set given more time. In regards to cell treatments, this graph exhibits the effect of oxidized albumin on IMR90s as seen by the red bars. And while there appears to be a reduction in cell viability relative to the albumin control, there are additional factors to be considered, such as heme, which could have been acting alone. So from here, I can only conclude that while this experiment is moving in the right direction, it requires additional adjustments for more optimized results. And so given additional time to dedicate towards this project, next steps would involve continuing to optimize a protein oxidation protocol, as mentioned, and continuing to assess both directly and indirectly heme damaged cells in the context of senescence. So including further testing for characteristics of aged and or damaged cells. And this would include SA beta gal activity, certain SASP factors, and increasing reactive oxygen species, for example. And by continuing to define heme-induced damage and doing this research and modifying potential solutions such as albumin, we'd be able to more accurately target the buildup and accumulation of this harmful molecule in the body and reduce its consequences not only in those suffering from various blood conditions, but in all of us experiencing the disease of aging. And to conclude, I just want to briefly express how the concept of ending aging or even delaying it has always seemed to me like a substantial and even inconceivable endeavor. And this is because we've been accustomed to believe that aging is a forever fixed fact of life, but who can blame us? And yet my time at Sense has shown me the value in altering that perspective and viewing the aging process as a disease that can be tackled like all others. And so I want to end by saying that the endeavor to minimize the risk of age-related disease and to improve our health spans and our lifespans in the long run is one very much worth the investment of time, effort, and optimism. And I'm very grateful to everyone at Sense for the opportunity to contribute to this effort and become even more excited about the future. And I'd just like to quickly thank and acknowledge the entire Sense lab for the mentorship and guidance in navigating this project, and the education department for this very rewarding experience, and everyone at Sense. So thank you.
Thank you so much, Danielle. I'm getting a flood of comments both through this platform and through, my email saying how much they appreciate your perspective and your reminder that aging is pathological and can be tackled. So I'm glad to hear that that paradigm is changing. It's fantastic to hear. We do have a couple questions in the chat. One of them being, how do the levels of free heme and albumin compare to the levels to which cells in solid organs would be exposed outside of hemolytic crisis conditions, such as having a stroke? Do you need me to repeat that?
Sure.
Okay. They're asking about the levels of free heme and albumin in solid organs in a normal environment compared to the levels of heme that you're treating cells with.
Right, I see. So I believe the concentrations that we were testing were matching the physiological conditions in a normal scenario. And I guess you could say a hemolytic event is a normal scenario. I mean, it happens in everyone, but yes, I do. I'm not completely certain that we that my project covered all the bases in terms of the different situations and different concentrations and ratios in different diseases and conditions. But yeah.
I understand completely, and unfortunately, summers are short, but
Right.
that this work is very exciting, and I'm excited to see where it leads as those comparisons are made. With that, I again just want to thank you for your time, and I'm going to turn it over back to Maria.
Wow, that was incredible.